In this video, we will be going over the sodium-potassium pump. There are 7 steps in total, and we will go over each step one by one. And so, starting off, here is the diagram of the sodium-potassium pump. In pink is the cell membrane. Above the cell membrane here, in this part, this is the extracellular side, meaning outside of the cell. And then below the cell membrane, in this area, we have the intracellular side, also known as the inside of the cell. And this in purple is our sodium-potassium pump. The green circles represent sodium ions. The red triangles represent potassium ions. And so we will go over each step or phase along with their description, the key event that happens in each step, as well as how it actually looks like in reality. So starting off with the first step, binding of sodium ions. The pump binds 3 sodium ions from the intracellular side of the membrane. So as we can see here, the pump is in its original natural conformation where it's open to the intracellular side and it's closed to the extracellular side. And so 3 sodium ions from inside the neuron bind to specific sites on the pump, which are represented in the circles. So here we see we have a sodium ion bound to the pump, sodium ion number 1, sodium ion number 2, and here we can see the third sodium ion is entering the specific site on the pump. And so the key note from this specific phase is that the pump binds 3 sodium ions from inside the cell. The second step will be phosphorylation. So as you guys have noticed, the sodium-potassium pump is now closed and it has the 3 sodium ions within it. And so for step 2, phosphorylation, ATP provides the energy for the pump to change shape. ATP is hydrolyzed, transferring a phosphate group to the pump, which induces a conformational change. And so here, the hydrolysis of ATP is represented with this arrow here, and the phosphate group is now attached to the pump. The phosphorylation or attachment of the phosphate group to the pump causes this shape that it's currently in. And so the key note here is that ATP is hydrolyzed, phosphorylating the pump and causing a conformational change. ADP right here and the free phosphate group are released. And also one more thing you guys can note is that the intracellular side has a very high concentration of potassium ions and the low concentration of sodium ions, whereas the extracellular side has a very high concentration of sodium ions and the low concentration of potassium ions. Moving on to the third step, we now have sodium ion release. So as you guys can see here, the pump is now open with the open side facing the extracellular side and the pump being closed to the intracellular side. And as we can see here, the sodium ions have now left the binding sites and have traveled on to the extracellular side. And so sodium ion release involves sodium ions being released outside the cell. The conformational change exposes sodium binding sites to the extracellular side, releasing the 3 sodium ions outside the cell. And the key thing to remember here is that the pump changes shape, exposing sodium binding sites to the extracellular side, and the pump releases 3 sodium ions to the outside of the cell. Step number 4, we have the binding of potassium ions. So now that the sodium ions were released, 3 of them, the pump is still facing the outside, and as you can see, it takes up potassium ion number 1 and potassium ion number 2, which we can see is entering the binding site with this arrow here. Another thing to keep in mind is that the pump is still phosphorylated. So number 4, binding of the potassium ions. The pump binds 2 potassium ions from the extracellular side of the membrane. 2 potassium ions from outside the neuron bind to specific sites on the pump. And the key note here is that the pump is still in its phosphorylated conformation, and the pump binds 2 potassium ions from outside the cell. Number 5, we have dephosphorylation. So as you guys can see here, the pump is now in its closed position with the 2 potassium ions still bound inside, and the phosphate group is now being released, which is what caused the pump to begin to revert back to its original shape. So 5, dephosphorylation. The phosphate group is released, restoring the pump's original shape. The pump releases the phosphate group, which triggers it to revert to its original conformation. And so the key note here is that the potassium ions are still bound and the phosphate group is released. Pump begins to revert to its original shape. A way I like to think of it is when the pump is dephosphorylated, it doesn't have any phosphate group attached, then it will be open towards the intracellular side. And if the pump is phosphorylated, it has the phosphate group attached, then it will be open to the extracellular side. For step number 6, we have the potassium ion release. So as you guys can notice, the phosphate group is now completely gone. The pump is fully dephosphorylated in the open position on the intracellular side. The 2 potassium ions have left the pump. And so the key thing here to remember is that the potassium ions are released inside the cell. The conformational change exposes potassium binding sites to the intracellular side, releasing the 2 potassium ions inside the cell. And so here, the pump released 2 potassium ions inside the cell. The pump returns to its original shape, ready to bind sodium ions again. And lastly, the cycle will repeat. The pump now is ready to start the process again. And just like in the first step, the pump binds 3 sodium ions from the inside, and the cycle continues to maintain the resting membrane potential and ion gradient.